working mechanisms of non-invasive neural stimulation on the brain in psychiatric illnesses, amongst them addiction.
Good. Esther, are you online? Can you open your video?
Yeah, yes, I'm online. I tried to open, but it didn't work yet. So, but I'm hearing you.
Could you introduce yourself?
Oh, yes. I'm Esther. I'm from Brazil. I'm from Federal University of Espírito Santo. We have in here working with a TDCS on the alcohol use disorder and also crack cocaine use disorder. So we have used multiple sessions, and we have seen some interesting results on craving control.
Good. Colleen.
Hi, I'm Colleen Hanlon. I'm an associate professor at Medical University of South Carolina in Charleston, South Carolina. I mainly study addiction. We're using mainly TMS in combination with functional MRI and structural MRI. I'm interested in, of course, like all of us, trying to use brain stimulation and our knowledge of neural circuitry to come up with a new treatment for addiction. But really, at the moment, I'm super interested in dosing and like optimizing dosing and tailoring it, frankly, figuring out why things don't work all the time.
Martin.
Hello, I'm from Germany, and I'm interested in brain stimulation in combination with psychotherapy, and mainly in the field of anxiety disorders. And here we try to modulate in the first step relevant processes for the therapeutic process, like fear extinction, and the next step we bring it into therapeutic trials. I did some studies with alcohol and craving before, but we have to wait some years now, and now the patients are here again, and we can add some studies for addiction too.
Thank you, Mark. Katie, could you introduce yourself?
Yeah, hello everyone. Good morning. Good morning my time. I'm Katie Witkiewitz. I'm professor of psychology at the University of New Mexico in Albuquerque, New Mexico in the US. And I'm primarily an addiction researcher, primarily alcohol, but also opioids and other drugs, and primarily behavioral treatment development for treating addiction. And got interested in brain stimulation about 5 years ago from a colleague, Vince Clark, who's here at UNM. And we embarked on a series of studies that I'll actually talk about today, so I won't go into that. But yeah, I'm very interested in what Colleen's working on as well, because we have a failed trial, and so I'm potentially leaving the field of brain stimulation and returning to my comforts of behavioral treatments that work well. But yeah, I'll tell you more about all that.
Great, great. Laurie.
Hi, I'm Laurie Zevertano from the Center for Addiction and Mental Health in Toronto. I'm an associate professor at the University of Toronto in the Department of Pharmacology, and I'm currently doing a clinical trial of TDCS in conjunction with varenicline for smoking.
Awesome. And others who have not introduced themselves, anybody else who would be interested to introduce him or herself? Primavera, Raha, Hosna, Azale, Ali Reza, I mean, any other, because as your kind of webcam is not on, so I would just, Primavera, do you want to talk?
Can you hear me? Okay, perfect. Hello, everybody. So my name is Primavera Spagnolo, and I'm a senior research fellow, 50% with the National Institute of Neurological Disorder and 50% with the National Institute of Drug Abuse. And so I've been working in the field of addiction for the last more or less 15 years. And since 2012, I started also, working with brain stimulation together with Mark Hallett. I don't know if some of you knows him, but it's one of the people that kind of put together this this intervention. And right now I'm working on two different projects. One is in Italy, and it is a randomized sham-controlled TMS study in patients with cocaine use disorder. And then we're about to start another study here at the NIDA in patients with opioid use disorder to look at the combined effect of brain stimulation and cognitive reappraisal of opioid use. And that's about everything.
That is great. Anybody else who is on phone or online who would be interested to introduce? No? Okay, good. So first of all, I would like to kind of let you know that we already have the last two webinars online on YouTube. So if you just kind of search for INTAM Webinar, you can find those webinars online. And I think that I was just thinking that as we are moving forward, if somebody wants to start to do a brain stimulation trial among drug users, I think that seeing those webinars, webinar 1, webinar 2, and now webinar 3, could give people a good idea about the challenges that we have in the field, maybe people who are doing research in the field, and how they might be able to move forward for different sorts of studies. So I think that kind of those webinars that are recorded and they're online, there are essentially really good resources for the community to be able to have a good idea about the quality standards in the field and how they might be able to start and move forward. And then today we are going to have 3 talks. Katie, Colleen, and I will discuss about different topics that I hope that they would be interesting for people. And then we will have a discussion at the end how we can move forward to the next steps. In terms of the INTAM consensus paper, I should tell you that we already have 73 authors from 17 different countries, and the draft is ready to submit. So hopefully we'll submit it tonight or tomorrow. So Colleen and I, we are still kind of doing the final kind of confirmation of the authors and the other kind of minor things. But basically the list of authors and the manuscript is ready to be submitted to neuroscience and biobehavioral reviews. So it's going to be submitted then. I hope that we will have kind of decent reviews for that, and then it will move forward for its publication. We might still think about how we can put that on bioRxiv as well to give people an early access to the material that we already have inside. That will be another discussion that we will have inside the INTAM leadership core. We will discuss about those things, but we basically have the manuscript ready to go. So that's kind of, it took a long time, something like 1 year of putting all these things together. And other than just kind of having that for 1 year, we received lots of really, really nice feedbacks from people. And even the last round, I received something like 30 different reviews and kind of sorts of kind of revisions in the entire manuscript, and different sort of comments that are, most of them were really, really good. So we updated the manuscript, and I hope that the final result would be interesting for people in the field. Any question about the consensus paper? No? Okay, good. And then,
I think we should, can we give a round of, can you hear me? I think we should give a round of applause to Hamed for all of his work.
Thank you very much.
That is like an incredibly difficult amount of work that you put into it. So thank you so much for your leadership. Thank you.
Thank you.
Just putting all those authors into the manuscript portal is going to be a nightmare. So we all owe you for that.
Yeah, I hope that the final result would be helpful for people who are kind of active in the field and who those would be newcomers in the field in the future. And then that I will talk about the INTAM quality control checklist in the next steps, kind of as the last author. Okay, so the first speaker would be Katie, and she will talk about negative findings in recent trials. So let me just, Katie, give you the screen.
Yeah, is it possible for me to share slides?
Yes, you can share your screen right now.
Okay, great. Excellent. So let me play this and see if this works.
Yes, we can see it really well.
You can see the slides?
Yep.
Excellent. Okay, I can't see you, but I can hear you, so that's good. So I will talk about kind of a program of research we tried over the last 5 years, actually, of my life. Not that I'm better. Trying to look at whether we could enhance the effects of mindfulness-based interventions with transcranial direct current stimulation. So I'm going to kind of walk you through our entire kind of the last 5 years and what we did, and then what we learned in that process, and where to go next. And some of this work is under review, some of it's actually in press. I'm happy to share papers and talk further with anyone interested. As we all know, neural system dysfunction and addiction is widespread. And so this is a really nice paper that's a few years old now by Yuan Tang that just kind of brings together the different models of neurobiological dysfunction and addiction and the different addiction cycle, and actually talks about mindfulness as one way of addressing this. And sure enough, there's a long literature on mindfulness meditation and brain adaptations with mindfulness. And of course, mindfulness meditation has been around for about 2,000 years or so. There's a lot of support for mindfulness generally, and a lot of good work kind of in using mindfulness to look at brain responses, mostly in healthy controls. I will say that. So as mindfulness is in the news all on the cover of Time magazine, so I don't have to go into like meditation works and mindfulness is effective. I used to have to spend 5 or 10 minutes convincing people that there is actually data to support mindfulness meditation, but I don't think I can do that anymore. There's been a lot of studies on meditation and mindfulness generally being associated with attentional control, physical health, immune function, greater self-awareness, greater self-regulation, reductions in perceived stress, and again, changes in brain structure and even function, even with just 8 weeks. So we developed several years ago now a treatment called mindfulness-based relapse prevention. It's very much based on mindfulness-based stress reduction as well as mindfulness-based cognitive therapy, and basically brings together mindfulness practices with kind of cognitive behavioral relapse prevention skills. It's delivered in a group over 8 weekly 2-hour sessions. Incorporates both formal and informal practices. I can talk further about that if anyone's interested, as well as kind of practicing at home. People do practice. That's a big question that I always get, but we do find that our patients actually practice meditation in between sessions. Those that practice do tend to have better long-term outcomes. The groups are led by mindfulness practitioners, people who actually have a practice, also who have experience in treating the underlying disorder that we're targeting. We've conducted 3 RCTs of mindfulness prevention for substance use disorder. All of these are published and now getting old, including our first study, which is now 10 years old, which is wild. But in each of these trials, we did randomization to different active treatment conditions. Of course, in all the trials, it was multiple active treatments and no placebo. Obviously, you don't tend to do placebo for behavioral interventions. In all the samples, they were diverse, both ethnically and racially, as well as diverse with respect to substances. So these are more pragmatic trials in that we're in the community and actually doing this work in the community. And what we found across all of these trials, and again, these papers are published, is that mindfulness prevention was more effective than treatment as usual, more effective than relapse prevention, and in our biggest trial, these effects were maintained up to a year. So the last trial of MDRP. You can see pretty large pre-post effect sizes and then smaller effect sizes for between treatments. So I'm just going to pause there. Are there any questions about the treatment or what we're doing in the treatment? Okay. I will then jump to why we moved into electrical stimulation. here. So, you know, these are really good findings, and we generally find that we're helping people. It's modestly efficacious, you know, effect sizes ranging from 0.17 to 0.41 versus an active treatment. And we do find that MBRT reduces craving and the relationship between depression symptoms and substance use. So it's kind of targeting what we want it to target. But treatment engagement and completion rate is low. And I don't mean low, but I mean lower than the alternative treatments. And you know, for those of you who've tried meditation, you know that it's not easy, right? Like sitting with your own thoughts can be quite difficult. And especially for people with addiction who might have already hampered prefrontal functioning, hampered in executive control, decision making. So the ability to kind of sit with and be with breathing or body scan or whatever meditation we're doing, it's just difficult. And so that's where we come to potentially augmenting mindfulness with stimulation. And so basically how this all started was Vince Clark at UNM was giving a talk on his work with transcranial direct current stimulation. And I was just blown away by the effects he was finding, obviously, and also this idea of enhancing what you're learning. And so I thought about what if we could enhance the learning of mindfulness. And so I think our work here, I will just say, is really different from a lot of the other applications of TDCS or TACS or TMS in addiction, in that in this work, we're not so much trying to tackle the addiction straight on. We're not tackling craving, we're not tackling any sort of addiction processes. What we're trying to do here is enhance learning of mindfulness. So the idea is that, you know, we are meditating during TDCS, and the TDCS can help focus the awareness on the breath or on the body or whatever we're focusing awareness on. And so the idea with meditation is that you're sitting, you're paying attention to your breath, your mind wanders, you bring your attention back to your breath, acknowledge that without judgment, and you do this, you know, over and over and over again. And so the idea was that TDCS could enhance this awareness in bringing people back to kind of their breath or their body, whatever they're focusing on. And I will say, you know, when Vince and I first started talking about this, I wanted to try it, and I was really blown away by how TDCS actually helped focus my practice, and not just in the moment of practicing. Practice for several days, my own meditation practice for several days after TDCS was kind of more solid. It was easier to bring my awareness to the focus of attention. So this idea, and maybe if someone could mute if you're talking there. So as far as a placement, and this is, you know, we're going to come back to this, but a lot of the work in addiction obviously uses, for those of you who know, DLPFC as a placement is pretty common. We were interested in the right inferior frontal gyrus, anodal stimulation of the right inferior frontal gyrus, mainly because of kind of the current modeling of potentially getting to insula, which we think is a key piece of meditation, is that self-awareness and insular involvement, but also just frontal cortex generally. And of course, it's a diffuse current flow. And so this is from Marome's group, showing the current distribution for right IFG anode and contralateral arm cathode. And one of my research assistants, we, in our latest trial, worked with Soterix and had these kind of custom headbands and armbands for the electrodes. And I'm going to just, if there's any questions about this, because I know it's different. Okay, quiet group. So moving in, so we did our first trial, and we do this in groups. So these are my research assistants at the time, and that's me, ironically, wearing the same exact scarf and necklace as I am right now. So, that's awesome. You know, I did not plan that. I wear this a lot. So we did weekly group. Our first study, we worked with smokers, and the reason being is that smokers are a little bit easier to find and generally want to quit. And so we had a very small amount of money. We actually ran this trial on $3,000. And so we had a very small amount of money. We wanted to do the work very quickly. And so we recruited smokers, and we recruited 9 smokers. They were 44% female, average age of 35. We did, MBRP is typically 8 weeks, but we thought with TDCS, we can shorten that to 4 because it's going to be so effective that we'll be able to cut down on the amount of treatment. And so what we're doing here is, so basically the first 30 minutes of the group session is a group meditation with TDCS. And in this first trial, it was just all active, no sham control, no blinding. Everyone knew they were getting active stim. We were just trying to see if there was any signal here. And so then what we do is at the end of that 30-minute stim at 2 milliamps, we took off the equipment and then did 60 minutes of the mindfulness training. So basically it's a 30-minute meditation practice during stim, take the stim equipment, 60 minutes of kind of smoking mindfulness intervention content. And so what we found in this initial trial was a large effect size reduction in cigarettes per smoking day at follow-up, so just at the end of treatment. And we also found a 33% biochemically confirmed smoking abstinence rate, which is pretty good for a 4-week intervention for not in the tobacco field. These folks were not getting any additional pharmacotherapy. This was just 4 weeks of this treatment, and 33% of them were abstinent by the end of those 4 weeks. We did also find medium effect size reductions in drinks per drinking day. So good effects in this initial trial. It made us feel pretty excited about moving forward. In the meantime, I had a student, Elizabeth McCallion, interested in using this in her dissertation, but she was interested in chronic pain. This paper is currently under review. And so we used TDCS in kind of a modified mindfulness-based stress reduction. Again, only 4 weeks for chronic pain. We assessed 91 people for eligibility. Average age was 68%. Average age was 37, 68% female. A broad range of pain conditions. And we had pretty good completion rate. So of the 47 randomized, 79% completed treatment in the active intervention and 74% in sham. In this trial, it was sham controlled, but it was single blind. So the participants didn't know which condition they were in, but the research assistants did. And so in this study, what we found is no main effects on mindfulness pain or overall health. But these 2 graphs on the left is, oops, sorry about that. On the left is mindfulness scores, and we found that for those in the active condition who attended more session, there was a significant effect of active stimulation on mindfulness scores, so it did seem to enhance mindful awareness. And then on the right is the same effect of active, the solid black line, for more groups attended, resulting in greater overall health. So the E5D, if you're not familiar with this measure, lower scores actually indicate better health. It's kind of a measure of dysfunction. Higher scores indicate worse health and more dysfunction. So some promising kind of, again, a little bit of a signal here. We're showing increases in mindfulness, improvements in health. I'm going to pause there.
Sorry for interruption. It seems that we have kind of colleagues that they already have their mic on. So would you just turn off your mic, because your mic could make a noise during Katie's presentation. So if you just kind of take a look inside your mic. Giovanni, I think that your mic is on. Martin, your mic is on. Laurie, I mean, those who have kind of their mic on, please kind of turn off your mic just to get sure that Katie would not have extra noise in the meeting room. Sorry, Katie, for interruption.
No, that's fine, Ahmed. But this is also a good time to pause to see if there's any questions either about the smoking trial or this chronic pain trial before I move into our big study.
I had a question, Katie. I was yes. kind of looking inside the picture that you have been showing. Have you been using the iontophoresis device for your pilot trial?
Yes, so that's a very good point and something I wanted to talk about as part of this, because I think it's actually a really important point. So yes, in the first two trials, the smoking trials and the chronic pain trials, we were using the Ekhtiari ion devices. And so these were obviously a lot more affordable, and for the pilot, both this pilot study and the dissertation, we were interested in more affordable devices. So that is also why in a group setting, for those of you who don't know, to double blind the Ekhtiari devices, you actually need 2 devices and a blinding box per person. And so, you know, with 9 people in a group, we would have needed 18 devices to double blind it, and that would have been really hard. Obviously, we didn't have that many devices, and it would just be a mess. I mean, it kind of, you can see in this picture, it kind of already looks like a mess with just, you know, here in this session, there's only 6 devices. But yeah, so that we were using the Ekhtiari in these initial trials. But that changed. Very good question.
I also have a question, Katie.
Sure. Go ahead, Colleen.
So why did you, just out of curiosity, why did you choose to do group-based therapy instead of just individual mindfulness?
Well, so it's always, the treatment's always been done in a group. We always have tested it in a group setting, and we feel the group setting's really important for, from a mindfulness meditation perspective, for sharing common challenges of the practices, for starting to recognize that kind of the ways that we approach life are very human ways, that we kind of have generally, you know, a lot of people experience anxiety and depression, and talking about those things in a group setting can normalize it. And so that's why we didn't want to make 2 changes. We didn't want to go to individual treatment and TDCS. We wanted to stick with, we know these behavioral interventions work in a group, so let's test them in a group. But I think that's a really important point. We're actually currently starting our first trial of MBRP as individual treatment without TDCS. So we'll know if it works for individuals, and that might be a next direction, is to do TDCS in individual settings. One of the things that does happen, that happened in the chronic pain trial and that happened in the heavy drinking trial, is that in a group setting, there is, because we're running people active and sham in the same group. And so the people, in talking about their experiences of the meditation, would mention aspects of the stimulation. And so there was a little bit of unblinding via discussion. More so, not so much the participants, but to the therapist. The therapist kind of would start to get a sense of who was getting active versus sham. So I think it's a really good question and another potential direction to go.
I have 2 small questions, if I may still.
Yes.
Once I was not 100% sure why the extracephalic cathode and not on the contralateral hemisphere or something. And secondly, about the combination of the TDCS and mindfulness training. Was it the TDCS during the entire session of mindfulness, let's say 60 minutes, or was it just only a part of it?
Yes, so great questions. So we went with the contralateral arm because we wanted to try to reach more subcortical. And so we were thinking that based on the current modeling, we didn't want the stimulation purely at the cortex. We wanted some subcortical if it gets there. We don't know, obviously, but that was the thinking. And also it was the montage that Vince Clark had previously used and we had effects with, and anecdotally people had experienced kind of this greater mindful awareness with the contralateral arm cathode. So that's why we stuck with that montage. As far as the program, so yeah, the first 30 minutes of the session is mindfulness with the stimulation, and then the rest of the session, there's 60 more minutes of the session, another hour. Actually, in the later trial, I'll talk about it, was another hour and a half of just intervention mindfulness without the stimulation. So it wasn't the entire session, just the first 30 minutes.
The reason that I ask that we are trying to do a TDCS group session, but with rumination, and we are discussing when we should start a TDCS. Is it at the beginning or in the end, when the mind is already set to the, like for instance, the mindfulness or the ruminative processes, which could influence your outcome results? But it's great to hear what your reasoning is. Thank you.
Yeah, yeah. Our thinking was that because there is, you know, studies have found that there is kind of carryover learning even after TDCS is shut off, that might enhance kind of the rest of the group. But I think it's an open question of whether beginning of group or end of group is a better time to do it. Okay, well, I will move into now our big study and lessons learned. So our next study was actually funded, so that's good. We actually had money to do a larger trial. So we did a double-blind, randomized, sham-controlled trial. We recruited drinkers in this study mostly because NIAAA was really interested in funding this work, and so we went to NIAAA and focused on heavy drinkers. That's also something I'm more kind of used to. It's kind of more my expertise. We did switch to the Soterix device, the mini CT device, which allowed us to do kind of double blinding in the device, and it was just much less onerous with respect to kind of having the system. You can see here my RAs, my graduate students, and they have, they're all hooked up, and now we have a nice meditation room as well. They're all hooked up and meditating here, pretending to meditate. And so each person kind of had their own little unit, and we had RAs who were monitoring, walking around, checking sensations, all of that. But it was weekly 2-hour sessions for 8 weeks. The first 30 minutes was guided meditation with right inferior frontal gyrus active TDCS versus sham. And then 90 minutes of MBRP content after we discontinued STIM. And so basically people would come in, we would have them do a brief set of questionnaires, we would hook them up to STIM, they would meditate and be stimulated for 30 minutes, we would discontinue STIM, and then the rest of the 90 minutes just be the MBRP treatment. So we recruited 84 heavy drinkers who are interested in reducing drinking. And I have the consort here, and I'm just gonna walk through this because we had to screen a lot of people to get to 84. And one thing that we decided to do in this trial, and you know, maybe it was a mistake, I don't know, but we were really cautious about prior history of alcohol withdrawal. And so we really didn't want, we're in a psychology setting, not a medical center. So we're on an academic, you know, main academic campus. We didn't want anyone having a seizure, and we just didn't know about seizure risk. And so we just decided for this initial trial to have people who had experience of alcohol withdrawal not be in the study. And that was our main exclusion. So we ended up with kind of a less severe drinking sample. Of other exclusions, metal in the body, obviously left-handed, people who didn't really want to reduce their drinking, basically who weren't really seeking treatment, any seizure history, current other substance use disorder other than cannabis, and we did end up having about 30% of our sample were cannabis users, and that's an issue. We then, what we did was we randomized pre-baseline, and the reason for that is we wanted to do a test stimulation session at baseline because we wanted to exclude anyone who just couldn't tolerate STIM. And so what we did was we randomized to active or sham. So we randomized 134 people, but a lot of people actually didn't come to the baseline, so they weren't included. So we ended up, our final sample sizes were 47 in the active intervention and 38 in sham, so 84 total. That doesn't quite add up, but I'll have to look at this again. So just kind of giving you the sense of the effort to get the sample. But then the sample itself, we did want to be, other than these exclusions for safety reasons, we did want to be inclusive. And so you can see our age range was 19 to 77. That could be an issue that we could discuss. The mean age, it was an older group. The mean age was 52. and the median age was 55. So it was an older, lighter drinking sample generally. The mix of ethnicity was consistent with the Albuquerque metro area. So about just under half were non-Hispanic white and 38% Hispanic, and then a mix of other races. Does anyone have any questions here? Because I know that there's a lot to unpack in this one big slide. Okay, I'll move on then. So we did do an intent to treat analysis, of course, and so even though you can see some people attended no groups and in both conditions, but that was twice as many inactive attended no groups, but we did do intent to treat. So all these individuals are included in the analysis. I will say that we also did a per protocol analysis where we analyzed the data for only people who attended more than one group, and we found no differences. So it really, the fact that we included people who attended 0 groups in an intent to treat analysis didn't really impact the final results, which were null. So this is drinks per drinking day. Over time, there was a 2-month follow-up. The red line is active and the blue line is sham. You might think that there's seemingly a trend, but there is no effect here. The effect size, I think, was 0.04. I should include it on the slide. It was very small for our primary outcome of drinks per drinking day. People did reduce generally, but not any differentially inactive. We did find a bit of a quadratic effect for mindful awareness. So there did seem to be, similar with the pain study, that individuals in the active treatment condition did seem to have greater mindful awareness at post-treatment, but that was not maintained at the 2-month follow-up. So I'm not too excited about this. We maybe did push up their mindfulness awareness a little bit, but that was not necessarily maintained beyond the treatment, which maybe speaks to the need for booster sessions, or, you know, the mini CT units are designed for kind of at-home use. And so maybe this is something that people would need to buy and have in their homes and kind of use regularly to help maintain their mindful awareness. So I just want to kind of give some conclusions across trials and then move into some of the big areas, I think, of where we made mistakes, what we learned. But one just thing I noticed in the course of these 5 years of investing in this approach is that as we increased the rigor, we see less effect of TDCS. And I think that's bearing out in the literature a little bit. We recently completed a systematic review of some of the TDCS. I know there's a lot of reviews out there right now, a lot that just came out. In fact, as we were working on the review, like a new paper would come out every month reviewing the literature of TDCS for addiction. And across the studies, you generally see that as the rigor increases, as you go to double-blind, sham-controlled, you see less effect of TDCS. And that's certainly what we observed in these 3 separate trials. You know, the first study, which was totally uncontrolled, we had big effects. And then the next trial, which was single-blind, sham-controlled, we were seeing some effects that continued to, you know, make us excited. And then in our double-blind, sham-controlled trial, we ended up with null findings. So, I mean, I think I'm walking away from this line of work with basically a lot more questions than answers. You know, with regard to stimulation parameters in particular, you know, the question about what about contralateral, you know, doing right and left anode cathode? What about dorsolateral prefrontal cortex instead of the right inferior frontal gyrus? These are a lot of things we don't know. Dose is also, I think, a really important one. We just went with 2 milliamps. And I really think, for example, with the Activa dose device, personally, 2 milliamps was really a sweet spot for me for mindful awareness for my own practice. When I tried mindfulness with the Soterix mini CT device, 2 milliamps felt too much, and 1.5 milliamps was a little bit of a softer kind of helped me be aware, but wasn't too painful and distracting for myself. So I think there might be both questions about dose with respect to individual differences of what people tolerate and what helps them, versus what actually gets medication by being so kind of painful and distracting. We certainly bring the stimulation into the meditation practice. So if you're noticing kind of enhanced sensations, to be aware of them. But still, I think, you know, if we're trying to enhance mindfulness, increasing awareness with maybe less of a dose for some people might be useful. And then, you know, we did 8 sessions. That's what the treatment is, but do we need more sessions? Do we need less sessions? The timing of the stimulation in the session, I think, is an open question. If I could do this all over again, I think I would have gone, you know, instead of doing a big RCT with just my 2 conditions, I think I would have done more of maybe a within subjects design, or an ABAB, or some sort of design where we could start to get some of these questions of what happens when we use DLPFC, what happens when we use 1.5, what happens with fewer sessions, what happens if the stimulation's at the end. So I think a lot of things we could have figured out before we launched into a pragmatic efficacy. And so I really, you know, wish I could take that back a little bit, but it is what it is. I think also our sample, there were issues there. You know, we ended up with a lighter drinking sample that was older, and also it was a study of people who were actively drinking. I've talked with Reina Weirs about some of his work in an inpatient unit, and we were talking about how maybe it's important for people to be abstinent in these trials, and I'm a harm reductionist, so I'm very much a fan of, you know, if people want to have a reduction goal, that's great, and so that's how we ran this trial, but it's certainly something that could have reduced the efficacy. And then there was, we did allow co-occurring cannabis and tobacco use in the study, and about 30% sample used one or the other or both. And so, you know, I think it's a question of, for me, it's always balancing external validity versus internal validity. And I weighted this trial more towards external validity, and I think that might have hurt us in our efficacy test. But it also makes me wonder if even if we found this to be really good in an internally valid trial, would it generalize to the community? I don't know, given it didn't work in this sample. So that's all I have. Happy to open it up for discussion and questions. And we made a lot of mistakes, and I recognize that. And you know, I hope you all can learn from our mistakes and maybe different direction. Or improve upon what we did. All in there.
Great.
Yeah, thank you. That's
great.
great.
Yeah, it was great. I have a question. Okay, I have a very feel very weird that why you use the IFG but not the DLPFC as the stimulant for the treatment the echo user. Use.
Yeah.
It's
So
very confused me, because, you know, most of study the previous is always use the DLPFC, not the inferior front of genius. Yeah.
And so again, so we were not interested in targeting the addiction processes themselves. We were not interested in targeting craving or addiction. We were interested in targeting mindfulness. And so we found in our preliminary work that the IFG placement was better. We did some preliminary testing of DLPFC. We also tried, one of my grad students did a big study with college students and looked at TPJ, temporal.
Yes, yeah. If you focus on on the mindfulness, I think the TPJ is a better areas than
In our preliminary work, it wasn't, and so we stuck with the IFG. And you know, I think again, I think it's open for it's an empirical question of whether TPJ would have been better or DLPFC would have been better. We tested IFG, and I can tell you the IFG wasn't better. So.
Okay.
Yeah.
So maybe I can add. I'm aware of a study in Belgium using mindfulness and TDCS, and then in let's say the classical setup with TDCS, and it's not published yet, but I've heard they didn't find any effect of TDCS, sham or real, on the mindfulness training as well. So this is a bit similar from another sample, another group sample, but I'm aware of one as such a study.
Yeah, you know, it's interesting interesting. I had a lot of conversations with people because I straddle the addiction research community and the mindfulness research community, and a lot of people in the community were pretty negative about the idea of kind of speeding up or making mindfulness like McMindfulness, making it like easier and for people, with the idea that it's a rigorous training and that it requires commitment and engagement with the practices, and practices are hard, and that's part of the practice. And so do we want to make it easier? Do we want to make it better for people to meditate? And so I think that's another interesting question with TDCS enhanced behavioral interventions. Are we in some ways shooting around enabling a person's efficacy for their ability to do things through their behavioral changes versus through stimulation? And we have people who said they were coming just for the stim, and so to the extent that maybe it gets people to come to treatment, that would be good, but I don't want people thinking that they just are getting better because of stimulation, not because of their own self-efficacy change.
Okay, another question, sorry.
Yeah.
Okay, you can first. Lady first.
Colleen, yeah.
So thank you so much, Katie. It's awesome. That was a really nice presentation you put together. It's really hard to show everyone data that you feel like it wasn't what you wanted to see. For sharing that, because we all have data like that. So I did have one science question, but before I ask that, so I would like to suggest, to reframe it, that I don't think you made mistakes. I think you did the best you could with the information that was available. And frankly, we just don't have a lot of good information available yet. So I think you did a good job, and we, like you said, we have so much room to learn about dosing and stuff. So
yeah. But
for your R21 study, so it looks like neither the just mindfulness alone and sham, and the group that got real, neither of them really changed their drinks per drinking day very much. Yeah. How did that affect compare to previous studies you've done in your lab with mindfulness without sham?
Yeah, so the pre to post effect size was a little bit smaller than in our previous studies. And so it was about, our previous studies have always been like 0.8 to 1 as far as pre-post effect sizes for mindfulness. And in this study, it was closer to like 0.4 or 5, I think, as far as reductions in drinking. But also, it's the first time we've tested this as an outpatient treatment for drinking reduction. So I think that, the fact that we got people who are already drinking less to begin with, we had a couple of people in this study who drank less than me, and I don't drink that much. And so I was like, wait, what? They're coming to treatment to reduce their drinking? I don't think wait, what? So I think it was a lighter drinking sample and less room to move. Our average drinks per drinking day at baseline was around 7 or 8, and in most of our trials, it's around, or in most clinical trials of alcohol use disorder, it's around 12 to 17. So as far as baseline drinks. So I think, having that alcohol withdrawal criterion really made our sample less severe to begin with, and just less room to grow. And so I think it speaks to a question around when you're thinking about inclusion criteria, recognizing that if you make it so that your sample is going to be really safe for TDCS, you might end up with a less severe group and less room to change.
Good.
Yeah.
Great. Katie, thank you very much. We need to move forward for the next talk. So we are because of the limited amount of time that we already have. If we can have time at the end, Dr. Zhang, we will have time to discuss about more details. So the next speaker would be Colleen, Dr. Hanlon. Would you move forward and just present the talk? We cannot hear you.
You're muted.
I'm just going to give you the screen. Just a second.
That's all. Okay, can you see my screen?
Yes.
Yes.
Okay.
Yes.
Great. So I'm actually going to be a talk. I am not as prepared as Katie. I didn't provide my own talk, but I So there's a clinical TMS society meeting that was in Vancouver in February, right before the International Brain Stimulation meeting. And there, Mark George gave one of the keynote talks, which I thought would be really valuable to share with our group. And so it's a little bit focused on what we've learned in terms of failure to find trials in depression. But I think it has a lot of relevance to what we're doing in addiction. So I'm going to go ahead and just say that these are mainly Dr. George's slides, and he graciously shared them with me so that I could share them with you. And so as some of you may know, there was a very big pivotal study that went on through the VA. It was a 9-site trial with the VA using TMS as a potential tool for depression. And it was published, I think, last year, and it basically showed no effect, so failure to find an effect. And so with that, there became a lot of interest and skepticism and whatnot over whether TMS is really having an effect. And that was the catalyst for Mark being invited to give this talk to the Clinical TMS Society meeting, which he titled, Recent Failure to Find Clinical TMS Trials. Are they natural growing pains in a field or something more problematic? So, so I think kind of in the good spirit of what Katie just told us, you know, it starts off this idea of you can learn great things from your mistakes when you aren't busy denying them. And I know that that's true myself too. When I see data and it just doesn't fit my hypothesis, you know, it's my first inclination is to think, oh, well, I must be doing something wrong. Let me think about it a different way. But in fact, like maybe that's just it. Maybe we were wrong. And sometimes that's hard, at least for me, to like remind myself that it's okay to be wrong, and it's okay that your hypothesis didn't work out, and we just have to learn from it. So what I'm going to tell you in the next sort of few minutes is our kind of problems with publication and presentation bias, keys to successful brain stimulation trials and movement to the market. This is from Mark George's perspective. I'll talk a little bit about the OPT-TMS trial, which was one of the FDA pivotal trials that got TMS approved for depression, as well as several, actually 3 recent failure to find TMS trials, which were multi-site trials that did not find their primary endpoints. And then we can talk a little bit about relationship to our field as an addiction researchers and the future. So positive publication bias, I think we're all very aware of this. It's receiving a lot of attention in the news and certainly at the NIH. And, you know, summarized by this little clever graphic here, we have published studies and then we have boring studies. And if any of you have tried to ever publish a study that is basically shows no significant difference, it's actually really hard to publish them. I myself have a set of data which I think is potentially one of the most rigorous sets of data that we've ever collected, and we see no effect. I've presented this at a few meetings, maybe some of you have seen it, but it's theta burst dosing. And frankly, we don't see an effective dose. We do 600 pulses, 1200, 18, 3600 pulses of CTBS, and we see no pattern of effects. And then we recently redone that with intermittent theta burst stimulation. This is over the motor cortex. And again, we find no consistent pattern of results. So one case, of course, is that we're doing something wrong. And that's, you know, that's certainly possible. So we try to be as rigorous as possible. And another case is just that it's not that simple. And what's been published before, the positive studies on theta burst stimulation over the motor cortex, they've just received a lot of press and attention. And so the field has moved on. And for us, it's actually very hard to publish these other studies. So that's probably affecting all of us, not just my lab, of course, but probably all of us out there. And sort of a caveat, another sort of element to that is, you know, as a PI running a lab, writing a paper takes a lot of time. You know, we get negative data sets a lot. It takes a lot of time and effort to compose a scholarly manuscript that integrates the field and tries to, you know, have some impact on our knowledge. Do we want to spend our time and effort on the studies that come out with no significant difference? Well, it tends that we also tend to get really excited about positive studies. But this unfortunately holds the field back, right? Because we don't know, we all reinvent the wheel all the time, and probably even those of us on this call, or certainly in INTAM at large, are replicating one another's failed studies. So hopefully, my hope is that through INTAM, and Hamid's doing such a great job leading us all together, we can, these webinars will help us as a community stop sort of reinventing the wheel. And we'll learn that, you know, Katie did this study and she was very rigorous using the information that she had available to us, and didn't find a very large effect size. Well, that's super valuable to us. So again, thanks for all of the people that have already shared their data on this webinar. I think it's great. So kind of to summarize, why is there a publication bias and a presentation bias? And this is in neurostimulation as well as in other fields. It's because frankly, we're more excited by positive findings. They're easier to write up. We kind of fool ourselves into thinking that it's much more likely to be causal, because we in fact had a solid conceptual framework, and then we evaluated the framework in a rigorous way and we found what we expected. And so it seems easy and causal. If we didn't, something is wrong. And then certainly positive studies are have an easier time in peer review. They're more likely to be cited. Dr. George said that he sees this even at brain stimulation as a journal. So, so what we're trying to kind of do is sort of help encourage people to share their negative results such that in an open way, such that we all can benefit. And also, honestly, even at meetings when you're invited to give talks, to sort of not be afraid to share your failure to find kind of things. Because again, it's just presumably we're all really good scientists. Just because you don't find your intended effect doesn't mean you're a bad scientist. Just means that science is hard. So this is Mark George's sort of view of the world. His sort of simple keys to a successful evolution of brain stimulation technology to market. So what he advises is to know the target, so the brain region that you're modulating. It's very common that we all just use a target that was used before us. And we just say that, well, Smith et al. Put the electrodes in this configuration, and therefore we are also going to put the electrodes in this configuration. But it would probably be more powerful if we knew a little bit more about the brain instead of just saying that we're doing it because somebody else has done it. Knowing which patients to enroll. Katie kind of talked a little bit about this, and this is going to be one of the reasons that people now think the VA pivotal trial just failed to find a large effect. As veterans are a really very unique group. Understanding the placebo effect, the effect size, at least with our TMS, certainly in our hands, the placebo effect is very high. And I think some interesting, if we ask people if they've got real and sham in our studies of alcohol users, cocaine users, opiate users, and nicotine users, most of the time people think that they're real TMS. And we have a pretty strong sham. So it's an active sham. But I think one of the interesting things about TMS, at least, and the placebo effect, at least guessing if you got real or sham, is that everybody gets a little bit of real, right? So you find the motor threshold in everybody. So everybody has that really novel salient experience of having the TMS coil placed over their motor cortex and their hand moving, and they didn't cause it to move. So that's really powerful. And whether you're in the real group or the placebo group, you get that. So that already is biasing our participants to perceive, to expecting that they're kind of getting some type of intervention. Knowing the dose and the time of the effect, this is really very hard. It's very hard to know how, as a field, we're going to parametrically go through all of the potential areas for dosing, but I'm optimistic that we will get there. Conducting multi-site trials that are adequately powered to find an effect. I think as Katie again elegantly showed, and this is true in science in general, when we have small studies, we often find really large effect sizes in either direction. But as you increase your power and you increase your rigor, and you basically control for a lot of the other variables that might be affecting, we lose the power. And that's not too unusual. That's not unique to neuromodulation. It's just science. And then his final point was to have a business plan that makes money for someone. You know, it seems funny, right? We're academic institutions, you know, there's some weird bias. We're not supposed to be contaminated by industry and all this stuff. But the truth is that it's industry that goes to the FDA. It's usually not individual investigators. And so learning to work with industry helps us try to figure out how to design a trial that's scientifically rigorous, but also has the inertia and the backing to move it forward. So I think in the addiction space, that's going to be an interesting thing for us to develop. There's a few TMS companies already that are starting to invest in the addiction space. It'd be great for all of us. But I think that it's kind of our responsibility to talk to them more than we normally do, and not to be afraid to talk to them, because the more that we communicate with industry, the more they will be well informed about the features that matter and design good studies, etc., which ultimately help our patients. So, Dr. George is sort of natural evolution of TMS, eventually on its way to an FDA-approved treatment for depression. And he cited in 1995, I believe he was at the NIH at this point, working with Mark Hallett, actually, and Bob Post. He was at NIMH and NINDS. And there he was in the graph on the top left here. He had an individual patient, and this patient's depression score is plotted over time for 90 days. And this individual received TMS over in these various intervals. You'll see here there's a phase 1, a phase 2, and a phase 3. This is a case report study published in 1995. This is, I think, one of the first studies that used RTMS as a potential tool to change depression scores. So that was in 95. In 97, they did the first sham-controlled trial. Again, I believe that was at the NIH. And they looked at active and sham TMS. It was a crossover design, but again, it was actually very small. And you can see that even it wasn't a huge effect. So from 97, it took a long time, over 10 years, until they actually executed and finished the multi-site study, in which they had randomized 190 individuals to receive real or sham TMS. That's a long time. So I think when when all of us hear like, oh, we're trying all this stuff and we're having negative results, we could give up. Katie, I hope you don't give up. We could give up, or frankly, we could just make the judicious decision that we want to pursue something else that's more fruitful. But the field, all fields need to mature, right? Like a baby. The baby doesn't know how to do anything at first, and they have to be shaped and optimized, and they're going to fall and they're going to trip. But eventually, they'll learn how to walk. And I think that's going to be true. It was true for depression. And we see now 10 years after the OpTMS study, depression field is still full of studies where they're trying to optimize the dose, and they're moving the coil around to different positions. They're sorting the field into biotypes. I actually would argue that they're a little bit behind some like substance use research and OCD in terms of most depression studies don't combine it with any type of behavioral intervention. It's just TMS alone. So the field now, 10 years after the FDA approval, is still refining its parameter space. So there's a lot of longevity in the field, but it's it requires that we sort through all of the sort of mess at first. And I think a great step in that place is for us to all begin talking and sharing what works and doesn't work. So those years kind of were critical for the ultimate success of TMS in the depression trials. In those 10 years, they learned to refine the dose. So they moved it up to 120% of the resting motor threshold. That was based on thinking about or learning about the distance between the scalp and the cortex. So as you move from the primary motor cortex towards the DLPFC, the space gets a lot wider. And correspondingly, you would need an elevated increase in the machine output in order to adjust for that dose. So now we're doing that in a much more sophisticated way, right? We can have automated segmentation, and we can automatically calculate the distance between the scalp and the cortex. Some of our neuronavigation devices will do it for us. So we're still continuing to refine that. In terms of depression, they really did do, so Holly Lusenby led a really nice trial. I think Paul Fitzgerald might have been involved in this, where they figured out how many sessions were required in order to have a stable dose, a stable effect. They worked on understanding the sham response and what's important to have a sham, what's the estimated effect size of an active sham, and to develop a true sham TMS in double-blind trials. So at least my pulse on the field is that TMS really does have a lot of good sham systems built in. The MagVenture device, for example, has a really nice double blinding system where you can flip the coil one way or the other. The BrainSway device also has a really nice system itself. The sham, in my opinion, needs to sort of be a little bit better. But the device companies basically are really kind of getting on board. Neurostar has their own sham TMS coil, which looks similar. So the device companies are helping us in this regard, which I think is another reason that we need to talk to them in order to help them understand what we need as scientists. So then last year in June, this big study came out in JAMA Psychiatry, got a ton of press. And this was part of a VA cooperative studies program. Began in 2004, so about 14 years before it was eventually published. And what they demonstrated, and I'll tell you what they demonstrated in a second, but their idea was to see across 9 VA sites if in fact TMS had efficacy to treat the veterans. And in this case, you can see here, it was an effectiveness trial. They asked the question, does TMS work to treat depression in US veterans who typically have past substance use, comorbid anxiety disorders, and medical problems? So these things like substance use and comorbid anxiety disorders were excluded in the TMS for depression trials. So, so the early studies had very clean samples, but now kind of we're moving the field into a more sort of normal sample that would come in this case, to a VA. The trial itself was a randomized parallel double-blind study with 9 clinical sites. They used the MagVenture figure 8 coil at 10 hertz, 120% motor threshold, 4,000 pulses per session, 30 sessions for 6 weeks. So that's like the standard, pretty much the standard depression protocol. It was nicely randomized, active and sham. The location was 6 centimeters from the motor hotspot. So, we now know that that's maybe not the optimal location. But again, trials, this is the challenge of getting into a big double-blind placebo-controlled trial, is that it takes a long time. And so all the information that we learn from the start to the end, is going to, we may be a little bit too premature because we may have a lot more discovery science to do before we know the optimal parameters. So this is Jerry Savage. This is he was the PI on the study. The study itself was initially powered for 300 individuals. 164 were randomized. The individuals were veterans from 18 to 80 years old. They had current their current episode of depression was less than 10 years. They were on psychotropic medications, and they excluded people with bipolar disorder, psychosis, or current substance abuse. But previous substance abuse, actually previous substance dependence was made people eligible. So what they found here, if you look at figure 2, is that if you look at the first graph here, A, individuals with major depressive disorder without PTSD, the dark bars are the active group and the light bars are the sham group. So the remission rate was between 40 and 50% in both the real group and the sham group. That was the end of the treatment, the main phase of the study. In the group of people that at the end of the follow-up study, the remission rate in the active was about 35% and the sham was about 25%. So in this case, there was no significant difference between real and sham. But what you notice here is actually 45% of people remitted with sham. That's really high. So the placebo rate in the study was exceedingly high. And so I think in kind of the post-mortem evaluation of what happened in this study, a lot of people have been wondering about why the placebo rate in this study was particularly high, higher than a lot of the other studies that have been done. And so when they look at this trial compared to some other trials, these are some things that have been mentioned. This was the first randomized controlled trial with TMS that added TMS to existing medications. In actual clinical practice of TMS for depression, patients are always given TMS on top of existing medications, at least here, and my knowledge of most centers. So in fact, the OPT-TMS clinical FDA pivotal trials really are not a very good indicator of what's going on in practices. Although medications weren't changed, there have been increased medication compliance. There have been issues with medication compliance during the trial. Many of the patients seem to sort of have a lot of esprit de corps associated with involving themselves in the trial. So I don't know if you guys have ever worked in the VA. The VA is such an interesting group of people, mainly men, and they really do have this tremendous like team spirit and loyalty. We recruit for some of our trials at the VA, and what you find, the research assistant goes and gives them a spiel in a group setting about our studies and asks them if they're interested in signing up. Then they can just sign up on a clipboard, and we'll give them a call. And we generally get buy-in from the whole group or no one in the group. And it's usually led by somebody in the group itself who is like a champion or a skeptic of what we're doing. And somebody speaks up and they'll act like the leader and people will follow them. It's so interesting. So just as in our own experience with the VA, there's this kind of loyalty groupthink. There's some idea that the esprit de corps of the veterans themselves make them over-report positive effects because they want to help their fellow man. These are some of the things that were mentioned in the JAMA report itself. Other VA trials, for example, have also suffered from really high placebo responses. I mean, if you're a veteran, that's great actually, right? But if you're a scientist, it makes it really hard to find a difference between your real and your sham. Does anyone have any questions? Okay, I'll keep going.
No, I've got a question.
Yeah.
I didn't read the paper, but what makes you sure that they over-report their positive effects?
Well, so anybody knows if they over-reported positive effects, but certainly the placebo rate was very high. So if you, in this image here, you can see that the placebo rate, so people that got sham TMS without PTSD, about, I guess, probably like 43% of them at the end of treatment had a remission rate.
Okay, and how do you think this is, because I heard you saying something about a group loyalty effect. How do you know this was not a group treatment, I suppose?
No, it was individual treatment, but they were all but there's an idea that people go to the VA and they sort of hang out in the VA. And so in this case, people they would come to their visits and they would talk to one another. And so they all, I think this is all speculation, so no one really knows this. But I think in the discussion section of the paper, they talk a little bit about this potential positive bias in VA trials in the past.
Okay, thank you.
Colleen, I do have a question.
Yeah.
Hi, this is Vera. So how much different is the placebo response from a pharmacological study or from DBS study in depression? Do you have an idea?
Yeah, that's a great question. Let me scroll back to the OpTMS study. So this is not the same data here, but this is a remission rate for the OpTMS study. The study design is a little bit different than the study that was done in the VA, but in this case, the remission rate among individuals that received sham was 5%. And the active rate, the response rate, was 14% with real TMS. So there's usually, there were probably 3 pivotal studies, I would say. Reardon did a big study, Lisenby did a big study, and they all show a pretty significant differentiation from sham. If anything, I think they're usually criticized because the actual TMS rates aren't really crazy high. And so they're better than sham, but the effect size isn't super high. In comparison, in the VA trial here, you'll see that the remission rate percentage, and again, this is a little bit different protocol, but the remission rate is almost 50% in the active group and about 45% or 43% in the sham group.
Because I do remember Ellen Mayberg when she presented her DBS, data in depression, talking about, like the very high placebo effect she was, seeing in her patients. And that brings me to the very last, very quick question. That is, were any off-target, outcome measures? I mean, and that also brings me to what Katie was discussing when she was saying her patients were cannabis or, users or smokers. Sometimes maybe to disentangle this question between the placebo effect and the real, effect, especially in this population of patients that tend to be, I would say every patient is actually susceptible to placebo effect, including me. But anyway, if I expect something, then of course, this expectation may affect, my response. But if I also look to some off-target effect, let's say, like in the case of Katie's study, cannabis rate consumption or something like that, then I may have a clearer idea of what's going on, if there is any effect there, besides what patients are told they should expect to, or the reason why they're, enrolling in the study. If I have depression, of course, I think I'm gonna see something about my depression happening.
Yeah. Yes, I think that's a perfect point. So they published, these are the main outcomes of the study that were published in 2018. I suspect that we'll be seeing some follow-up papers in the next year or two about some secondary effects or off-target effects, as you suggest. I think one thing that was pointed out here in this figure is that you can see, so the presence of PTSD had a large effect on the effect size. So that's one thing that they pointed out. So individuals that are individuals with major depressive disorder without PTSD. Those that had PTSD had a much lower remission rate, whether they were an active or sham. So I actually think that's pretty interesting because separately, remembering this is 10 hertz TMS to the left dorsolateral prefrontal cortex. Separately, there have been VA studies, I'm not sure if they were multi-site or single-site, studies evaluating 10 hertz DLPFC TMS as a treatment tool for PTSD. So these results would suggest that PTSD interfered with or decreased the effects of both real and sham. But that, which is, potentially interesting. If you would think that if 10 Hertz TMS was also an effective treatment tool for PTSD, that maybe the depression and the PTSD would both get better, or certainly that PTSD wouldn't be a confounding effect in the results. But it just seems that the story is not that simple. Okay.
Thank you.
Yeah, thanks. So, so if we kind of come back to what he pointed out about these kind of rules of knowing the target or the brain region. So they use the one that's been used before. There's now, there's pretty good functional connectivity and brain structure to inform that. Knowing which patients to enroll, understanding the placebo effect size, conducting multi-site trials that are adequately powered to find the effect. I'm not sure why they didn't finish the trial. So if you remember earlier, they had intended to enroll about 300 individuals, but they only enrolled 167 or something. So they didn't actually enroll the full amount. So that was a little bit of a challenge. But this, as I said, the idea of a failure to find effect has been shown in a lot of other VA cooperative studies. So this might be something unique about the VA protocol. So a recent study by John Crystal evaluated naltrexone as a treatment for alcohol. I guess it's not really recent, but and in that case, they also had a relatively high sham and real, or placebo and real effect. That was true for sertraline for PTSD in the VA, antipsychotics for schizophrenia and a depot formulation, risperidone, and prazosin for insomnia. So again, some of these issues may be related to this particular population. So then kind of to move on, I don't want to take up too much time, but to move on to the next thing, another study published in 2011 also was a failure to find study. And that was in adolescents for depression. They had 100 depressed patients from 12 to 21 years old. And they haven't actually published the final data yet. So if you look back, they initially, it was published here in 2011 as the trial was starting. Neuronetics, I guess Mark George emailed Neuronetics about this right before the meeting in Vancouver, and the science officer at Neuronetics said that Neuronetics is currently completing the analysis of all phases of this study, including the randomized controlled trial, open label extension, and the 6-month follow-up study. Based on a preliminary analysis, the primary endpoint in the first phase of the trial was not met. Neuronetics will share the data with speakers after the analyses are complete. So we don't know exactly what's happened with that study, but it suggests that, again, this is a failure to find study. Hopefully we'll learn more about that. But if you look, it's 100 depressed individuals. It's pretty good. It's randomized and sham controlled. And so I guess some of the idea there is that according to the communication between Karen Hart and Mark George, is that maybe the placebo effect was relatively high in those individuals. And then finally, one last study was to discuss is the randomized controlled trial of navigated rTMS in motor recovery for stroke. In this case, they had 1 Hertz active or sham TMS in healthy contralesional motor cortex. So if you're not familiar with stroke recovery, one of the primary science issues in stroke is that you have a lesion in one hemisphere, and then in the initial stages of stroke recovery, your contralesional hemisphere increases as activity, and the amount of activity in the contralesional hemisphere in the early stages of stroke is associated with a high likelihood of recovery. But after after a few months, when you become in the subacute phase, about 3 months to 12 months after the event, activity in the contralesional hemisphere seems to interfere with motor recovery. So a common paradigm in stroke recovery research with TMS is to try to decrease activity in this contralesional hemisphere, trying to force the lesion hemisphere to be active. This is the same idea they use in constraint-induced movement therapy for stroke recovery. So in this study, they used 1 Hertz active or sham TMS to the healthy contralesional motor cortex. So trying to inhibit the good hemisphere. They did 18 60-minute sessions over 6 weeks. So that's a lot of studies. They enrolled 199 individuals with stroke. 167 individuals completed the full 18 sessions. And that's a lot for people that have a stroke, which impairs their movement. And what they found was that everyone improved. And there was no significant difference between the groups. So again, we have another situation here where our placebo effect or our sham effect is really high. So I think in addiction, we're going to have to think more about that as well. I don't have a lot of time, so I won't go through this, but you all can see these slides on your own. So this is what the authors wrote in their report. Maybe the sham stimulation was actually active. Maybe the underlying rehabilitation that individuals were having while they were receiving the 1 Hertz TMS was more effective than we thought. That would be like in line with kind of Katie's comments about mindfulness therapy, which has historically actually had really good effect sizes, and is now considered a good potential treatment for various addictions. Maybe in fact the effect size of the intervention, the behavioral intervention, is strong enough that our brain stimulation intervention can't push it around very much. And then in stroke, one of the primary issues is lesion location. I think in addiction, this idea of lesion location is going to be important too. As we know, with addiction, we have a lot, especially alcohol, there's a lot of structural pathology that happens in the brain. Right? There's a lot of atrophy that happens. And when you have gray matter atrophy, you're going to get an increasing distance between the scalp to the cortex. And that should influence the amount of TMS or TDCS that's received at the cortical location. That'll be probably particularly, that'll be relevant for both of those technologies. But at least in our lab, we've shown that of all the various factors that could influence our effect size, like age and length of use and all kinds of factors, scalp to cortex distance is the most powerful. So it's very important. Another thing that we have been thinking a lot about recently is the role of white matter integrity from the source of the TMS coil to our target brain region. And we published a study last year, and we have another one under review currently, where we demonstrate that the ability to modulate bulge signal in subcortical regions like the caudate and the putamen, which we're really interested in addiction, is dependent upon the integrity of the white matter from the location of our coil, which in our case is the medial prefrontal cortex, and the subcortical region itself. So again, kind of like just in stroke, the cortical geography itself is probably going to be an important factor that affects our treatment outcome. And again, these are all sort of the same things, knowing the target brain region, which patients to enroll, understanding the placebo effect, the dose and the time, and then really trying to eventually conduct multi-site trials that are adequately powered. And so I think I want to just kind of stop there and get your perspective on things. But when I kind of saw that presentation and I thought to myself, how does this apply to us as addiction researchers? I think to myself, kind of like what Katie was mentioning, it's tough, right? There's a lot of factors. Dosing, there's like 1,000 review papers out of TMS and addiction or TDCS and addiction, and everybody says, how many sessions? What's the, total amplitude, all the standard stuff. So it's easy to just give up. But I think the preclinical basis is very strong, that stimulating certain neural circuits changes behavior. And it can change drug self-administration behavior. It can change cognitive performance like set shifting and animal behavior tasks. It can change things like delayed match to sample. There's like really great studies. So I feel like the conceptual basis is strong enough to make me sort of want to keep playing in this space. But I recognize, you know, we're not going to make a lot of progress. It blows my mind that 10 hertz TMS to the left DLPFC was one of the first things they tried, and it works. Because it's hard to imagine that for addiction, we're just going to come up with a magic thing like that. So that's all. Thank you.
Thank you very much, Colleen.
Thank you.
It was a really, really good presentation. And I think that that's exactly true. I mean, what we are supposed to do, we are responsible for the methods that we are using and how much kind of rigor we are putting in the study methodology, but we are not responsible for the results. So that's how us as scientists are basically supposed to work. And I think that in the kind of following that discussion in terms of how we might be able to increase the quality of research, that is basically why we are getting together, discussing about different things, how we can increase the quality of research independent to what would be the result. And we are supposed to kind of report whatever we have. And I'm really kind of happy that Katie is presenting what, I mean, she has done, and she has done a really, really nice study, and I really like it. So that's I think that to be honest, I really like it more than a positive trial. Yeah, there are many occasions that I basically learn a lot more from negative trials compared to positive trials. So I think that would be a strategy that we kind of decided to push forward for INTAM, to kind of ask those who are brave and intelligent enough to put out their negative results and just help us to learn about that. And that would be great. So we will try to kind of keep on doing this. So if you know others that they are willing to report their negative results and they would be happy to discuss about different things that they are doing, I think that would be really invaluable in terms of kind of discussing about the details. And I was thinking about the trial that Katie has done in terms of even using different sorts of devices. And I have heard from different labs, because we have the same issue here right now, we have been using iontophoresis devices before, and we had a larger effect. And now we move to new, fancier devices, because when you do iontophoresis, the DC device that iontophoresis would provide would be really something like a sawtooth. So it would be something like there's a DC, but over the DC you have something like a noise. But the new devices would basically provide a really clean DC. Could that be an effect? That might be an issue. Or what most of the devices that you use right now, they provide a significant amount of stimulation in their automatic sham protocol. For example, Neurocon device, if you do a 20 minutes of a stimulation for the real stimulation, and you activate your automatic sham stimulation, it would basically provide something like 40 seconds of active stimulation. It would go something like 30 seconds up, 40 seconds of a stimulation, and 30 seconds down. And then it would also measure in something like a 5 hertz recording over the time, with injecting a little bit of current during the sham stimulation in the entire amount of kind of the sham. So is that really a sham stimulation, or is this kind of could be considered kind of an active stimulation? And that could be the case for also kind of esoteric devices. So those kind of minor things might make a significant kind of difference. And I was wondering those things, how those things could be important, even for the field of like TMS. Right now, we know that different companies, the shape of the parts that they make are not exactly the same. Some of the companies right now, they make sharper kind of pulses, and some of the companies that they make, I mean, and there are occasions that we do not report those really minor differences in terms of even the kind of the whites of the TMS pulse. So these are the things that we basically need to discuss, and it would kind of go into the details in terms of how we can have something like a quality control checklist. Do we need a quality control checklist in the field to get sure that we are considering all these factors? And that quality control checklist should be updated rigorously over a time to just get sure that whenever we learn something new, we will add to the checklist to just get sure that others would also be kind of careful about how they are considering those factors in their study design. So let me just kind of share you my slides and start from here. Okay, so that is that's a kind of a question that we are discussing during last a couple of weeks with Colleen and other colleagues discussing about, and with Katie, we have been discussing, do we need a quality control checklist or not? And you know that in the field of medicine, we even have guidelines on how to generate these sorts of guidelines or checklists. So, I mean, the field is kind of moving forward in terms of providing outcomes, how we can do these quality control checklists. And then we already have a few specific checklists in the field of, let's say, quantitative research in psychology, or we have specific kind of additional checklist to consort checklist with specifically for psychological intervention trials. So we have kind of attempts in the field to bring quality control checklists to the field of psychology, or even we already have a few attempts to bring quality control checklists to the field of addiction medicine in terms of designing a trial or reporting a trial. And I think that Katie has done a really nice job, especially for alcohol use disorder, in terms of what are the kind of recommendations and checklists that people should consider. Specifically in the field of brain stimulation, the only major quality control checklist that we found so far is what people have provided in the field of TMS and motor cortex, how we can do kind of motor cortex stimulation with TMS, mainly single pulse and paired pulse kind of studies, and the international consensus study that is being published in 2012. And if I kind of give you an idea about what they have done, they had 2 rounds of expert panels. So they had 32 expert panels from different disciplines, and they kind of generated a checklist, and they asked people basically to answer 2 major questions. First, how much that factor is important to be controlled in a study. And then the next question would be, should people report that or not in their kind of their study? And for each item, let's say for the age of subjects, they basically kind of measure that with their expert panel. And the IQD is interquartile kind of deviation, and it would show the lower it would be, the higher consensus people made. So, I mean, it basically shows that the deviation of kind of reports that people provided. And they try to bring items that would have something like the IQD less than 1. So, but you see are most of the time are kind of less than 1. And then the percent of response for very important or always or most of the time. So the extreme responses should be higher than 60%. To include some of the factors inside. So as you can see that they have kind of included different sorts of subject factors, different sorts of methodological factors in terms of, let's say, for example, what would be the size of the call, what are the factors that people need to consider and report. And interestingly, even there are factors that they decided to remove from the checklist. There are factors there that I would like to be reported in my studies, at least. For example, the time of the day that we do the testing, and we know that if we do the testing early in the morning or testing early kind of in the afternoon, there would be a difference. So we know that these factors would be important, or amount of the caffeine intake or nicotine intake in the day of testing. But these are the factors that they decided based on the consensus not to put in the checklist at the end of the day, based on the threshold that they considered for their method. And that could be something that we can even discuss in terms of kind of what would be the threshold of including something inside. And I told you there are factors there that I really like to include when I kind of write a paper, and I think that would basically increase the quality in the field as well. So that is basically their reported checklist, and they ask people to even provide that checklist when they submit the manuscript. It's really hard to say that how much people are using the checklist, because you can imagine you can develop a checklist, but you do not have, let's say, a specific regulation in a way that forcing people to do something. So it's I mean, you can imagine that how much it could increase the quality of kind of activities in the field, but sometimes people, I mean, it's just a volunteer contribution to the field. And meanwhile, you can see that in most of the other checklists that people develop, they try to bring different journal editors across the world and across the field to publish that checklist to get sure that people or different journals would ask authors to consider those factors. So that would be another issue in terms of how much people would be happy to use the checklist at the end of the day. But besides all these things, the question would be, do we really need a quality control checklist? And if we need a quality control checklist, what are the factors that we need to consider there? So you can imagine that in a quality control checklist, there would be basically at least 3 major groups of items. One item would be things that are related to, I mean, general sort of clinical trials that kind of basically any clinical trial should be considered, should be considering those factors. And then there would be specific factors related to the different sort of non-invasive brain stimulation interventions. And then another category of factors that would be related to, let's say, field of addiction. And there could also be sort of interaction between those things that are important for the field of non-invasive brain stimulation when we consider drug addiction and so on. So there could be also some sort of interaction as well. So how do we really need to have a checklist in the field, or how this checklist is going to be helpful in the field or not? So I think that would be nice if you kind of share your ideas about that, and then I will show you a few other things that we have done for kind of the checklist as well.
So I think if nothing else, NIDA program would like one. So I think they, yeah, I mean, I think I don't know how we feel about that, but I think they would like a way to try to help with rigor and reproducibility in studies. I think there's probably some low-hanging fruit which everybody could agree upon should be included, at least in our TMS studies, and I'm sure the same is true of TDCS studies. You should report the location, the amplitude, the task they were doing, blah, 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 blah. So I think there's probably some easy things we could do, which might help people that are kind of new to the field. There's probably a ton of people that are addiction researchers that are never, ever, ever going to stumble on Siemens' work in clinical neuropsychology, right, or neurophysiology. So we could provide them with kind of a basic kind of checklist of things like, make sure you do all these things and you consider these things in your paper.
Mm-hmm.
Yeah, I agree. And I also think, I mean, when you first brought this up, Hamed, I think we don't need to recreate the wheel. I mean, the consort and other guidelines that are out there are really good. And I don't think we need to talk about those general kind of how do you do a clinical trial and reporting requirements for clinical trials. I think we need to really focus on those kind of specific things that are really important to report for a TDCS trial or DCS, TMS, TACS, whatever, electrical stimulation trial. And I think that would be really well received, but I just don't see the need to recreate all the other general components.
And if I can add something, I will say that maybe, I mean, considering like a target engagement study versus like a larger clinical trial, I will see that, the checklist, therefore the requirement could be slightly different. So for example, we have spoken a lot about location. So if I'm doing a target engagement study in all the meaning of target engagement, then saying I'm stimulating the left dorsolateral prefrontal cortex, personally to me doesn't mean that much, because the dorsolateral prefrontal cortex is huge, so where exactly are you stimulating? While, if I'm doing, a very large clinical trial such as at the VA, well, then it will be unlikely that, you can actually have 300 subjects, undergoing a baseline scan. I mean, maybe you can do a structural scan, but not a resting state or a task, and extract the big voxel and use that as your target. So I will say that, especially for target engagement study, the issue of the location, at least to me, it's really important, especially in the perspective of reproducing the data, because if we stick with the just left or right dorsolateral prefrontal cortex, then of course it's very difficult to replicate the data. If, for example, I'm using the 6 centimeters, from the motor threshold, another person is using the, EEG placement, or another person is using the anatomical, coordinates, you're basically stimulating different areas, and you're stimulating, therefore, different networks, because you really don't know where you're stimulating. And I think this is a very important issue, especially in the early stage of a study or of an hypothesis-driven study.
Any other comment? Anyone else who wants to kind of add something to that, or just be against of having a checklist and making people's life harder in the field? Okay, yeah, I think that's kind of what Primavera has mentioned is going to be also an important issue in terms of what, I mean, when you have a target engagement trial in an experimental setting compared to a clinical setting, there could be factors in terms of what would the level of precision in terms of the localization of the electrodes, or I mean, that could be an issue in terms of what would be the precision of reporting. But at the end of the day, we want to do whatever we want to do is supposed to be replicable. So even in the large scale trials, and if they are doing, let's say, kind of DLPFC, with just 6 centimeters from the hot motor spot, it should be a method that could be reported in a way that the people can replicate as it has been done in other sites. So that would be an important issue. But how?
Well, I can just add something very quickly that also, bring another issue, that is, we should agree, and that is related to the checklist, with what we consider like the minimum, safety requirement. So if we all agree that everybody that will undergo TMS or TDC needs a structural scan, because we want to be sure there are not brain lesion, etc., then the requirement of using at least anatomical, landmark, come together with, the safety requirements, right? Because you're going to have that data regardless that, you're in the VA or anywhere else. If instead we say, well, like the structural scan is optional, then of course, you had a further layer of complexity, because certain people can say, well, I don't have the data, so I just have to use the 5-6 centimeters. And I honestly think that, especially for the issue that Colleen was saying, the level of atrophy, that you can have in certain patients, with, that, with addiction, having at least a structural scan, to me, in my experience, it's very important, because sometimes patients, do not report what happened to them, not because they want to hide it, but just because they didn't think it was important, and then you look at their structural scan and you're like, okay, I don't think here, we can really do TMS or anything else right now. We have something else that is more important. So, incorporating in the checklist also something regarding safety could be, useful for, for but, the safety purposes in itself, but also for the localization, requirements. I don't know.
The challenge is, at least in the consensus paper, the discussions that we had with different people for the consensus paper, in terms of the safety, when we talk about safety and we want to add to what we already have in terms of the general guidelines of safety for TMS and TDCS, and we want to make that more complex for the field of substance use disorder, that would be something that we probably would not like to pursue to that level, because the field is already complex and we already receive lots of kind of feedbacks from IRB people, and we are not going to, because we know that it's not going to, we know that there are factors that we need to consider, but making the field kind of more complex in terms of the safety issue, probably safety would be a really complex issue in terms of how we want to add kind of complexity to the safety guidelines, specifically for addiction medicine. That is going to be a hard discussion with people who are kind of moving forward for that. But meanwhile, adding things like kind of requiring people to report or having structural scans, or even considering structural scan for their safety process, that is going to be also complex as well. So I think there are factors that could help a lot in the field without too much pain. For example, I was reviewing kind of most of the papers in the field when they use basically 5x7 electrodes over the head. And it's quite interesting that many of the studies, they have not reported the direction of the electrode, whether they have put the electrodes from this side or from the other side. So, and right now there are studies from different teams. Right now, I think Michael Nietzsche has recently published something showing that kind of direction would be important in terms of how would be the effect of TDCS. So there are really kind of minor things that people are doing in the field, but they are not reporting. So those things would be much easier to ask people to include in their reports, but just asking them, for example, to do a structural scan for any stimulation and consider it as a part of their, let's say, screening process, that could be a kind of a hard discussion. So if you have, Colleen or Katie or others, if you have any other comment on that, I would be happy to discuss about that as well. Yeah, so you can imagine that having a consensus on a checklist is not going to be easy, but it is a kind of an interesting discussion to see how we can move forward for that. Our current kind of decision is just to wait to have our consensus paper out as the first step, just having a consensus of people around, and then thinking about how we can move forward for the quality control checklist. But what we already have in terms of a quality control checklist, we have developed an initial checklist with let me just show you what we already have. We already have an initial checklist that we developed with Katie and Colleen and a few other colleagues that we have been discussing in terms of what are the major factors that should be considered in terms of the addiction-specific factors, general factors that others are considering for trials, and how they are going to be important, specifically for the field of brain stimulation for addiction medicine. And also specific things regarding the stimulation protocol, blinding status, standard environment. And there are specific factors that are going to be more important. Let's say, what would be the contextual intervention that we add on the stimulation to that, and how much detail we need to provide for that contextual treatment. So there are factors that definitely would be interesting to be discussed, and we are basically working on that. So we will start to share the checklist with others to see how we can move forward for the next step. One issue that might be interesting to follow as well is that we realize that we need to also have a specific collaborative network on specific methodologies in the field. For example, right now we know that it seems that we need to have a specific collaborative network on doing TDCS-TS with fMRI, because there are specific methodological issues that are related to that specific method. And that could be another approach to the field. Instead of just getting focused to addiction medicine and brain stimulation, we can think about different domains. And interestingly, even we do not have right now a really good checklist for TDCS or TMS trials. So that could be another level that we can discuss, how we might be able to have even a more general checklist together. And I've been discussing about that with other colleagues, and I think that people are interested to have even a specific methodological checklist, not only for the field of addiction medicine, but also for specific types of technologies that we are using. So we have already started an international network of people who are doing TDCS, TES, and fMRI together, and we had a webinar a few weeks ago. So those who are interested, they can follow that line, and they can kind of find the webinar on YouTube. And we are kind of pushing forward for that specific checklist as well in the field. But how the field is supposed to move forward? We hope that our methodologies are going to be replicable in the future. We hope that at the end of the day, we can pre-register the details of analysis that we want to do. And those who have already done pre-registration, they know that it is really hard, especially when you have, let's say, imaging outcome measures that we have different sorts of kind of contrast, we can have different sorts of analysis. It's really complex. And even for simple behavior analysis, when you have different time points of assessments, they are going to be complex when you want to pre-register everything. And how we might want to move forward and use something like Open Science Framework, that will be an important issue in the future. And then how we might be able to have online data collection settings and registration platforms like what we use in REDCap. How can we kind of do the online syncing of a data collection across different labs? And then how we might be able to share outcome measures in collaborative partnership with different labs, and then how we can share protocols and data collection platforms inside this network, how we might be able to have something like a matrix of research. For example, as Katie has been mentioning today, she is suggesting us to do some specific sorts of kind of fields of investigation in the future that she thinks they would be low-hanging fruits in the future to be explored. How we might be able to develop that matrix to be able to inform others who wants to run a new study to use that specific low-hanging fruit to be explored, and then providing the results for us in the community to be able to plan for the next steps, and how we might be able to have multicentric trials. These are the kind of the big ambitions that we have for this network, hopefully in the next couple of years, to see how we are able to collaborate on these sorts of partnerships in the future. So that is what we try to do, and we hope that we will be able to move forward for these collaborative efforts. So kind of we are 1 minute kind of behind the time. So if probably for the next 2 minutes, if you have any question or any thing that you like to discuss, then we can wrap up the meeting.
I have to go. Thank you all.
Yeah,
Good.
thank you so much.
Good. So have a super nice day for those who are in US and those who are in Europe or China or other places of the world. Have a good night.